I hope it's okay. We'll interrupt you with questions during the.
Yes, of course. I think that would be the best if you interrupt me. That's perfectly fine. I would like to start by thanking the organizer for inviting me to present my work here, and of course for inviting me to be in person over there. But unfortunately, I couldn't be there in person, and I appreciate the flexibility to allow me to present this over Zoom. So today I'm going to be talking about cell motility. By cell, I mean eukaryotic cells and also bacteria, with the emphasis of how cells may use the environment to change the way they move. And I'm going to present two different cases. The first case is how cells use cellular footprints. This is the work I've been doing during these last 2 years that finally we got accepted at PNAS last week. And the other part of the talk is going to be about bacterial adhesion to surfaces. And this is the work I did during my previous postdoc at the University of Nice. So why cell motility? So cell motility is essential in life. It's involved in many different processes in living systems. So for example, here on top, we have some examples of eukaryotic cells, motility and migration. So we have here wound healing, where cells are going to start moving to close a wound. So motility is also important during development, as we can see this very nice orchestrated and synchronized movement of many cells together that are going to finally define the differentiation of the cell and development of the embryo. So motility is also important in our immune system. So this is the very famous movie of a neutrophil chasing a bacteria. And also, cell motility plays an important role during cancer and cancer metastasis, where tumor cells invade healthy tissues. And motility is also important for bacteria. In the case of bacteria, so bacterial motility allows bacteria to move toward food sources or move away harmful scenarios, providing survival to the bacteria. And here also we have the swarming, this collective behavior of swarming of bacteria, where bacteria are sliding to each other to produce this type of interesting movement that is very important during biofilm formation, and also to form this fruiting body that happen when these bacteria start to starve. And of course, bacterial motility is also very important for bacterial infection. During infection, bacteria need to search and locate target sites to start infection, and for that they need to swim. But cells are not alone, they are not isolated. They live in very different media and different environments that condition the way that they behave. And these environments can be very complex, and they can also change over time. As we can see here on the left, the case of an epithelial cell that is crawling over the extracellular matrix. And you can see that the cells, as it moves, is going to pull over the fibers and deform the network. And these changes over the network sometimes are elastic, but some other times are more permanent, and they can cause changes in the properties of the extracellular matrix, like the topology or the stiffness. And those changes, of course, are going to affect the way that the cells behave. On the other hand, we have the case of Salmonella infections. So in order to cause an infection, Salmonella have to swim from the gut lumen towards the epithelium wall. And during this path, the bacteria has to traverse a very complex environment that is going to be full of different type of microorganisms and other cells. And you can think about this as obstacles. And it's also going to traverse the mucus layer that has a very different viscosity than the lumen. So we can see through these examples that there is a dynamic relationship between cell motility and the environment, where cells, they change their environment as they move, and these changes on the environment can produce further changes in the way that the cells behave. And this is what I'm interested in. I'm interested in understanding the interplay between the motility and the environment. And in particular, in this talk, the question I would like to address is how cells use the environment to control the way they move. And to address this question, I'm going to present this first case that is about how cells secrete footprint and can use these footprints to change their motility behavior. And this work has been inspired by this paper from Joseph D'Alessandro and Benoit Ladoux, in which they perform these experiments in which they fabricate these linear tracks that were coated with fibronectin. So fibronectin is kind of like a glue and a sticky glue, so cells can only stick to the fibronectin, so can only stick to the linear tracks, and they cannot go outside the tracks. So then they place MDCK epithelial cells over this track, and they observe them for a very long time. And what they found was something a bit surprising. So cells, instead of moving randomly over the track, they move in a much more deterministic way. So you can see that cells oscillate over time. And you can also see that these oscillations increase their amplitude over time. And the explanation that they found for this oscillation was that these cells are depositing some kind of chemical over the substrate and using this chemical as a footprint. And these cells are going to move in a different way depending on if they are over the footprint or not. So these cells move persistently when they are crawling over the footprint, but when they reach the edge of the footprint, that means a place where the cell hasn't been yet, now the cell is going to stop and turn around and start moving on the other direction. And it's going to keep moving until it reaches the other edge of the footprint. And then again, it's going to stop, turn around, and start moving toward the other edge. So this cell, so basically that's how we can understand this oscillation. And the increase in amplitude can be explained because every time that the cell reach one of these footprint boundary, during the process of turning around, is going to extend the footprint a bit further away. So this brings me to the question that I'm interested in here, that is how cells can use this footprint to produce these oscillations. And if cells can use footprint to produce the oscillation, can they use footprint to produce another type of motility behavior? And if so, how can cells control these? And another interesting question is, why hasn't anyone seen this before? So people have been observing cells for many, many years, but this is the first time that someone reports cells using footprints. And more broadly, if footprint can change the way that cells behave, it would be interesting to understand what is the role of cellular footprint in cell migration. And for example, we can ask the question, can cells leave a footprint so other cells can follow them using, of course, the footprint? So to try to address these questions, we built a model in which the cell secretes the footprint, and then the footprint is going to affect the way that the cell behaves. So this model has three components: a component that describes the cell motion, a second component that describes basically how cells fabricate and deposit this footprint over the substrate, and the last component that is going to describe how the footprint affects the cell behavior.
Just a question. So cells normally move in 3 dimensions or in 2 dimensions?
So in this case, it's just 2-dimension.
In 3 dimensions?
Normally, they would move over the extracellular matrix that is a 3-dimension problem. So yes, this is a simplification where we are just assuming that the cell is moving over the substrate in 2D, basically.
Yes.
I'm sorry, can you repeat the question, please?
3-dimensional version of a footprint, or does the footprint only exist if it moves in 2 dimensions?
You can think about a 3-dimensional footprint in the case of on 3D, for example, in the case that cells can type of deform and leave a path over the extracellular matrix. So like building like a hole over the extracellular matrix, that could be think about like a footprint in 3D. But as I said before, footprint, this paper from D'Alessandro and Benoit Ledoux is the first one that reports cellular footprint. So this has been only observed in 2D.
I see. Okay, thanks.
Yeah, you're welcome. So as I was saying, here we model the cell shape and motion using a phase field approach. So in the phase field approach, the cell is described by a field that takes the value 1 in those regions that are inside the cell and 0 outside the cell. And then we can define the cell boundary as where the field is equal to 1/2. And then the field is going to evolve following this equation over here that in principle has 2 terms. And the first term is what drives the motility of the cell. And that is, in this case, is driven by this polarity protein that we call RAC. So this protein basically what does is accumulate at the front of the cell and is going to push the boundary of the front forward. So this is the extension of the cell or the protrusion of the cell. The second
sorry,
term,
Emiliano.
yes.
Quick question: is this a continuous version of the cellular pods model or something like that, or?
Oh, you can think about this like a continuous version of the pod model. Yes, but this is a bit better than the cellular pod model, because in the cellular pod model, I might make some comments after about the cellular pod model, but or maybe on the next talk. But in the cellular pod model, you have the problem that you cannot track time in a very controlled way. So time in the cellular pod model is something a bit more abstract that has to do with your Monte Carlo steps. So here you can track them in a normal way, and also allows you to describe this continuously. So in that sense, it's a bit better to describe the shape and motion of the cell, but it's much more costly computationally. So that's why in some cases we go back to the cellular pod model.
Excuse me, another question. Phi, it's discrete or continuous?
So the field is continuous.
Oh, okay. So I have, I mean, I assume that, let's say, the transition from 1 to 0 are sharp, or I mean, how do I interpret the intermediate values?
So the transition, as you say, is very sharp, and we want that because that is going to define the boundary.
Right.
And that is actually controlled by this parameter epsilon. Or I don't know, yeah, I think it's epsilon, this how you call this parameter. So basically by changing that parameter, you can make the interface larger or smaller, but the transition happened quite fast. So
okay.
you will see this in the movies of the simulations.
Okay, thanks.
How do you keep going from 0 to 1? So
how you keep what?
Why can't phi become negative? It looks like this equation doesn't fix phi to be between 0 and 1, so.
So so you have phi is going to follow this. I was just saying this, follow this minimization of energy. And in this minimization of energy, you have this double wall potential.
No, you wrote down an evolution equation for phi.
I'm having trouble hearing. Okay, but in principle, let me say this. In principle, yes, phi can take negative values. There is no restriction to that. So you have to be very careful how you choose the parameters of these simulations to make it work. So it doesn't work with any type of parameter. So you have to be very careful of how you set up everything. And yes, you can have sometimes result that doesn't make any sense. But the way it works is that 5 is not going to take any negative value here. And that is kind of controlled by this Hamiltonian here. And the reason why the cell goes to 1 or 0 is because we have this double wall potential that is going to try to minimize. So and the solution are, of course, of 0 and 1. And then at the same time, this field is trying to minimize this linear tension in this model. So you can also ask other type of minimization. So for example, we are not asking to minimize volume, for example, because here volume is free. So the cell can extend the volume or reduce the volume.
Okay.
So, and let's say, if I understand correctly, this double well potential, the idea is that you want to minimize H, and so you minimize by putting phi equal to 0 or 1 because okay, I see. And I mean,
Exactly,
the choice of, I mean, just a stupid curiosity, the choice of 18, it's because you wanted something large compared to all the other scales, or
I don't remember exactly where it comes as 18. I don't think it plays any important role here.
No, I was curious because you have several parameters, alpha, beta, epsilon, and so on, that you, let's say, you tune, and then there is this 18 that looks,
you're completely right. And there is no important role of that 18
but you
there.
don't tune it. You take it for, okay.
Yeah, this is taken from the original paper where they use this value, but I think that 18 makes sense with the other parameters that they are using. So you can rescale everything. So that 18 doesn't play any important role. So what is important is that the minimum of this function is in phi equal to 1 and phi equal to 0. Okay, is there any other question? Okay, so I will continue. And okay, so the other important thing here is how what is driving the motility of the cell. So as I said, this is described by the concentration of this polarity protein that we call RAC. And this protein can be in induced state, can be in an active state that is going to be bound to the membrane of the cell and is going to accumulate at the front. But it can also be in an active state that is all distributed all along the cytosol. And so the dynamic of this active RAC is described by this equation. So it can diffuse, but also we have this reaction term that describes how active RAC become inactive and the other way. And the important thing here is that we have this positive feedback mechanism given by this term over here. And this is what allows the cell to sustain a polarity. So when when we have some accumulation of active RAC out the front, that accumulation is going to trigger more accumulation of RAC at the front. So in that sense, this positive feedback is what allows cells to sustain a polarity and to sustain a directionality in the movement over time. So now we add the footprint dynamic, and for that we are assuming something very simple. The footprint that is described by this F cell is going to be secreted under the cell with a given secretion rate, and of course the footprint can degrade over time. And the other important thing to note here is that we use this effective footprint response, and the reason for that is cells, they don't respond directly to the footprint over the substrate, but we assume that they are going to respond through some type of pathway where they are going to have receptors over the membrane that are going to bind the footprint over the substrate. So that's going to trigger a cascade of event inducing the response of the cell. So we use this response function that has this kind of Hill equation form, as you can see here.
Sorry, you're just working in 2 dimensions. So R is just it's X and Y, R.
Yes.
So is there a reason why you don't do the 1-dimensional case? Is the 1-dimensional case the same complexity as the 2-dimensional case, or?
So I'm going to present a model where we use a 1-dimensional case. The reason that we use a 2-dimensional case is because the shape of the cell may play an important role here. We lost the shape of the cell is we use a single a 1D linear model, and you will see that when how that is important when we present simulations of cells moving onto the substrate.
Okay.
But I'm going to present a simple model on 1D.
Sorry, related to this, I'm getting a bit confused here. In the, let's say, in the picture that you have on the left up, the 2-dimensional is essentially the black line, and it is, let's say, entering in the screen, right? This is the 2-dimensional
Yes.
space.
Yes,
So,
yes.
but there is also a vertical dimension, right? Where the green dots are, in other words, is the cell allowed to, let's say, to blow in the vertical or no?
No, I'm sorry. That is like I think that this cartoon might be misleading. So there is no C dimension here. So there is no height of the cell. So everything is projected over the substrate, basically.
Okay, so let's say the concentration of the green are supposed to be on the same line of the black one.
Yes, yes. Everything happening on that plane.
Okay, okay.
I have a cartoon that might be better to explain that. The only reason that I draw it like this is to show that there is a footprint being deposited under, but I understand that can be a bit misleading. So finally, we need to say how the footprint is going to affect the cellular motility. And we assume a very simple case, a very simple dynamic here, where the footprint is going to promote this positive feedback mechanism, as you can see here in this arrow. So the footprint is going to enter in this term over here. So you can see here this phi function that is basically the effective footprint response. So to understand how this works, let me give you this example. So this is the 2D representation of the cell. So when the cell is moving over a substrate where there has been a footprint, since there is a footprint, now this positive feedback term is going to be on. And so that means that the cell is going to be able to sustain a polarity, and that means that the cell is going to be able to keep moving in the same direction that was coming. But when the cell reaches a region where it hasn't been before, so that means there is no footprint, now there is nothing that is sustaining this positive feedback. So that means that the cell is going to eventually lose the polarity and is going to stop moving. Okay.
The cell size, right? There's some area of the cell.
I couldn't hear very well, sorry.
The cell has a finite area. Right?
Yes, has a finite area, yes.
Some part of the cell might be on a footprint, some part may not. I mean,
Yes.
I don't know what means.
That means that if we go back to this equation over here, so this row represents like the active rack. So we're basically the active rack is going to be accumulating where there is footprint. So basically this rho is going to be high if there is a footprint, but this also depends on the position. So basically, what is happening is that cell is pushing over those regions. So the cell extend its boundary on those regions where is an active rack, where is the footprint. So where is the footprint, the cell can extend the membrane. So this is given by this term over here. So that's why we have the gradient of the field over here. And we have this alpha that is kind of like the rate of protrusion, and we have this beta that is kind of the rate of retraction. But this can only happen if we have active rack. If we don't have active rack, this cannot happen, and we cannot have active rack if we don't have footprint, basically.
Sorry, when the full cell is on the footprint, then let's say this rack is more or less homogeneously, let's say, covering the full cell, right? Or not, or accumulating one there. Cioè, here, if you want, I mean, suppose that the cell is moving to the right. When I see that F is accumulating on the right and not on the left, if both are on the footprint?
Oh, because this has a previous history, no? So there is you can think about like a fluctuation that is going to produce a polarity at one end of the cell, and once that you have some accumulation of some fluctuation that accumulates,
this trigger more, okay.
This trigger more, and then once that the front forms, that's going to be sustained because of this positive feedback.
I see. And so when you exit, let's say this part becomes 0, and then how the accumulation on the other side? It happens because, as Nathan was saying, the other side is still on the footprint. So if there is a small, let's say, thermal is probably not the correct word, but let's say random accumulation on the left, that become positively sustained by the footprint.
Exactly that, and I can show you that in our model results. So I think that if I show you some of the movies, that everything is going to start making more sense.
Mm-hmm.
So we start by basically mimicking the experimental conditions. So we made our 1D track that we coded with kind of an initial chemical that allows the cell to only remain over there. And here what we observe, so this is the cell. The line represents the border of the cell, the edge of the cell, and the yellow color represents the activity of rack, the active rack. Okay, so you can see how this cell is polarized, and this polarity accumulates at the front. And you can see also that the cell is oscillating, and you will see that this oscillation increases their amplitude over time. Here on the right, we are seeing the footprint. So you can see how there is a footprint in all those places where the cell has been before. And you can see also how the cell is extending the footprint every time that it hits the footprint boundary. Okay? And we can make a zoom on one of these turning around events when the cell hit the boundary. So you can see how the cell is arriving to the edge of the footprint, is polarized and is moving with a given velocity. And once it hits the boundary, now it's going to shrink and slow down. And since there is no footprint at the front, then the polarity at that front is going to be lost. And since there is more footprint on the other side, then the cell is going to polarize in that direction. And then that front on the other side is going to win, and so the cell is going to start moving away. And this is something that was observed during the experiments, and we were not planning on this, but we were pretty happy to be able to observe that our cells are kind of reproducing the same that what was observed in the experiments.
One thing that I might be wrong, but that I noticed I was already noticing in the very first movie that you showed. I get the feeling that in the real experiment, let's say after a few oscillations, the cell was not coming back to the original, was remaining bigger in some sense. Is this reproduced or not? Or maybe I just misunderstood.
No, I think you're right with that. So initially the cells in the experiment start like kind of rounded cell and small, and over time the cells start kind of extending more and more. And so you can see that the volume or the size of the cell is larger at the end. We don't have any size control in our model, so that our cell can extend, but it's not reproducing exactly the way that the cells in the experiment change their volume. But you can see how our cell gets larger and extended when it's moving, and shrinks when it hits the boundary.
Yeah.
Okay. So we were able to reproduce the oscillations, but we also wanted to explore a bit more the interaction with the footprint. So it seems that one of the important parameters here is the velocity that the cell is able to build the footprint. So this is the secretion rate. So now I'm going to show you a movie where the secretion rate is much higher than before. So as you can see in this case, now the cell doesn't oscillate and just move persistently. And okay, so there is no oscillation, and this cell is able to move toward region that hasn't been before without any problem and keep moving. And the reason of that is this cell is secreting so much footprint that it's able to build the necessary substrate to keep moving on their way. And we have a cool cartoon here that kind of represents the idea. Oh, I'm so sorry about that. So this kind of like is the same idea. So the cell is building the footprint as it goes. And this is only allowed because the cell is secreting so much footprint. So we have this kind of two types of motility behaviors, oscillations and persistent motion. And we have these transitions that are kind of controlled by how fast the cell is able to secrete the footprint, but also with this KD parameter, that's basically what defines the amount of footprint the cell needs to respond. So this is kind of the buffer of the footprint that the cell needs to respond. And so we can build this transition phase diagram between oscillation and persistent motion, and we can see that the transition happen over this line where the secretion rate is proportional to this KD parameter. So far on all those movies that I showed you before, there was no degradation. So now we add the degradation into our simulations. And what we observe is that, yeah, in this case, we also see oscillations, but you will see that these oscillations, they don't increase their amplitude. So they are right to stay amplitude and stay over there. And the reason for that is that now, as you can see here on the right, when the cell tried to go back to the previous edge, the footprint got degraded, so there is no longer footprint over there, so it has to rebuild it. So that kind of confines how far the cell can go. And so that is what we observe. So basically, when you add degradation, the cell is going to start oscillating until you reach a steady-state oscillation. And this steady-state oscillation this amplitude, depends on how fast the footprint degrades. So the faster the footprint degrades, the smaller the amplitudes are going to be, and the more confined the cell is going to be. So we can think about this degradation as a way of confining the cell into a region, and that confinement can change by changing the degradation rate of the footprint. Okay, so as it was pointed out before, why to use a 2D model to explain a 1D phenomenon? And we thought about that, and also because the phase field model has make very nice movies, but it's very hard to make analytical predictions. So we went back and we make this very simple 1D toy model in which we have our cell that is moving over a lattice, and this cell follows deterministic rules. So the cell is moving to the right, and at each time it's going to ask the question, is the footprint larger than 0.5? If the answer is yes, it's going to move to the right. If the answer is no, it's going to move to the previous side. So to illustrate how this works, this is an example where there is footprint equal to 1 all over this red region, and there is no footprint otherwise. And so let me play from the beginning. So at the beginning, this cell in this position is moving to the right. Ask the question, is the footprint larger than 0.5? The answer is yes, then the cell moves to the right. Ask the question again, the answer is going to be the same, keep moving to the right. It's going to keep moving to the right until we reach this static site over here where there is no footprint. When that happens, now the cell is going to change the direction. And it's going to keep asking the question until it reach again the other edge, and again it's going to turn around. So this cell switch direction every time that it hits the boundary of the footprint here. So of course we want to add the footprint dynamics, so that's what we did. So footprint is going to be secreted with a given secretion rate and also degrades. And we want to use the model to understand the steady state condition. And to think about that, so to understand the steady state condition, think about a cell that is starting at the boundary of these steady state oscillations. So at that boundary, at that age, the amount of footprint that the cell secrete in order to sustain the steady state has to be equal to the amount of footprint that gets degrades over the time that the cell goes to the other edge and come back. So that is the condition that we need to ask to sustain the steady state oscillations. So this is what we wrote over here. And from this equation, we can obtain an expression for the steady state amplitude. And now we can use this expression to try to understand some of the results that we observed before. And we were pretty happy because this simple model can recapitulate some of the observations that we saw before. We can predict how the steady state amplitude dependent with the degradation rate. But not only that, we can also predict where this transition from oscillation to persistent motion are going to happen. And these are given by this denominator. So when this denominator becomes negative, we have the transition to persistent motion. And that is when this K plus parameter, so where this denominator is, numerator is larger than, is smaller than, no, it's larger than the denominator KT. And we can obtain this proportionality between secretion rate and this KD parameter. Okay, so we saw that 1D model can recapitulate some of the observations with our more complex model in 1D. But what happened in 2D? So now we code a square surface and place our cells over there, and we observe what happened. So here on the left, we have the low deposition rate case. So in this case is the analogous case of the oscillations on the 1D linear track. But now we don't see oscillation, but we see that the cell is moving in circles. And we can see that over time, these circles are expanding the radius. So this is very similar to what happened in 1D, where we have the increase on amplitude of the oscillations. Here we see that this is moving in expanding circles.
Sorry, I'm sorry. So I have two questions. First
Yes.
of all, is an experiment in 2 dimensions?
I'm going to go there.
Okay. I guess the other question is, what is making the thing turn? Why doesn't it just keep up the direction?
Why isn't keep moving?
Why doesn't, I would have thought naively that in 2 dimensions, if you just set it in one direction, it's just going to see the one direction. What is making it turn? Why is it angular momentum?
So that is a very good question. And we were also kind of puzzled about that. I don't think I have a very simple answer to that. I think what happened is basically the dynamic of repolarization given by this RAC activity of RAC equation make easier for the cell to change a bit the polarity front instead of repolarizing the whole cell in the opposite direction. So it's much simpler for the cell to turn around than just to repolarize completely in the opposite direction and start going backward.
All right, is your model deterministic?
No. So there is some noise in our model. So the noise is in the activity of RAC basically. So RAC the concentration of RAC has some white noise over the concentration.
Okay. Sorry, to substantiate this idea that, I mean, to me it looks reasonable that instead of, let's say, plugging back all the concentration, you just start to turn and then that creates. Have you tried to remove the noise? Because I would expect that if that is the right interpretation, once you remove the noise, you should see perfect circle, I would say. Have you tried to run the simulation without any noise and see what shape? Because here, I mean, it's not a circle, there is a curvature.
Yes, there is a curvature. No, we haven't tried that, removing the noise. We tried going on the opposite direction, trying to increase the noise, trying to see if we have a spontaneous repolarization of the cell. So our cells have some noise, but the noise is kind of small. We don't see spontaneous repolarization. That is something that you would observe in real experiment. So
No,
we have noise. The noise is small.
Noise. Let's say the rate of movement of the concentration should be constant, so probably you should see a perfect circle. So yeah,
I would imagine that yes, that might be possible to happen. We will have to check. I think that the noise is nice. I think it's quite nice that having noise and that this circle is not perfect. And actually, I think it's quite nice that the cell sometimes is moving over the edge of the footprint here, the edge of the white area, but sometimes it's just going to escape the edges and start wandering inside that region. So that is quite interesting from the point of view of diffusivity or mean square displacement. I don't know how I'm going to present a bit about this, but this make a bit complicated to understand the diffusion properties of this case over time.
and let's say so to say the radius of this circle grows forever, or you reach a certain saturation radius.
So that I think is an interesting question. Our simulations suggest that it will keep growing, but I would say at the same time that our simulations are not that long, just because of they are very costly computationally. So we cannot keep simulating this for decades and decades. So it might happen that it reaches to a point that it cannot keep moving. But for the moment, it doesn't seem to be that way. It just I'm going to show some mean square displacement plot later.
All right. Does your cell have only one place where it can So the cell that you're drawing here only has a head; it has no tail. So it always the head is always the place that's in front, right?
Yes, but the head can change. So the head basically is where the polarity accumulates, and the polarity, the active RAC can accumulate at this front. But if the cell loses the polarity, at some point it can repolarize all over the other region. But that is something that is not happening. So as I said before, it's just easier for the cell to move the polarity from to one side than just repolarizing on the opposite side.
That's what you decided, but I'm asking, in real life, does the cell have a head, or does it have two heads, or how many heads does it have?
The cell doesn't have any head. So in real life, cells can have multiple front or competing front. It can be polarized into one front. It can have, as I said, multiple front at the same time, and at the end, one is going to win. And and what is the front at one moment might not be the front at other moment. So what you define as a front later might front and back later can be back and front. So
That's
the
sorry,
polite
2 heads. I'm asking 10 heads a cell, or how many heads does it have? I'm just in real life. How many?
I would say that cell only have one head in general, but as
No,
I
but-
said,
Oh.
cell.
So it has 2 heads at least.
So maybe I can draw this. Can I draw here? Yeah. So in real life cells, we would draw a cell like this. Here would be the nucleus. So this would be the front. So this is the kind of picture that we can imagine of a cell. But on real experiment, you would see cells that are maybe like this and has another front that is growing. So you can have competing front, and at the end, one of these is going to win. And sometimes the cell extended in this direction, and at the same time extended in this direction. So it can have many competing fronts, basically.
Okay, so I guess you're going to tell us experiments, but you're going to say in 2. So in 1 dimension, certainly it needs both. It needs 2 directions, right? It can't oscillate. It has 1 direction, right? One can't oscillate if it's in 1 dimension if it only has 1 head.
I'm not sure if I understand it. So
1 dimension, if it oscillates, it keeps changing directions, right? First
yes.
it's one, it's the other head, then it's 1 head, then it's the other head.
Yes.
Okay, but you claim in 2 dimensions, there's always only 1 head. That's the claim?
No, that's not the claim.
Relation.
And I don't, so to be honest, I don't think about this as heads. So in principle, the cell can polarize in any direction. It's just that in 1D, it's constrained just to be able to polarize in one direction or the other direction. In 2D, it can polarize in any direction. So what is happening here is once that the front form, it's really hard for the cell to lose that front and repolarize in a new direction. But it rather change the direction of that front. So you can think that it's not that the cell is changing the head, but the front is just moving, shifting to one's direction, to the right or to the left, if you want.
Okay, thanks.
Okay, so this was the low deposition rate. So this is kind of the analogous case of the oscillations in 1D on 2D. But what happened when we increase the secretion rate? So this is the analogous case of the persistent motion in 1D. So now we see that our cell is persistent and it can start moving over the 2D region, even though there is no footprint. But we also see this, that when the cell run into the previous footprint, now instead of keep moving in the same direction, it might prefer to it might prefer to change the moving direction and to start moving over the previous footprint. Okay, and we can see that now a loop form, and the cell now is kind of looping around. But now we will see that the cell makes a different direction and start moving in a different way. So we have this kind of interesting combination of a cell that is able to explore the space, but at the same time has a preference to remain using the previous path and being confined or looping over the previous trajectory. So we have this kind of exploratory behavior with some kind of loosely confinement given by the footprint. So what happened in 2D? Okay, so we went back to D'Alessandro and Benoit Ledoux, and we asked them, do you have trajectories of cells in 2D? And they said yes. So they sent us the trajectories. So according to our model, since in 1D cells were oscillating, that was compatible with the idea that cells have a low secretion rate. So that means, according to our model, that in 2D we should be expecting expanding circles. And in fact, we observe expanding circles. This is a trajectory of the cell that is expanding, is an expanding circle. But this was actually a rare case. Most of the cases where cells that look much more exploratory and having this kind of overlap with previous paths. So this is kind of suggesting the other direction. So this is kind of suggesting that cells should be over the transition rate and they should be behaving kind of more like in the persistent way in 1D. So how could it be? Why our model is not predicting this? So we went back to our model and we found that what is happening is that the transition between 1D and between persistent motion and oscillatory motion or circular motion to exploratory motion happen at different places in 1D and 2D. So that allows us basically to have different type of combinations. So we could have, for example, cells that oscillate in 1D and cells that move in circles on 2D. So that was what we were expecting at the beginning. But we can also find a situation where we have cells that are oscillating in 1D, but they move rather exploratory in 2D. So all this is kind of suggesting that our cells should be in this regime where they are kind of slightly above the transition. Arguing kind of in favor of this is the fact that we actually see both type of trajectories. So we observe circular motion and we also see exploratory motion. And we can explain that we observe both of these because of cell-to-cell variability. So if cells are kind of slightly above the transition, cell-to-cell variability may pose some of the cells below the transition point so they can behave as circular. So lastly, as I mentioned before, I was going to show some mean-square displacement. So here on the right, on the left, we have the simulations, and here on the right, we have the experiment. Yes.
You think there are two different kinds of parameters for two different kinds of cells? You don't think it's just random noise that tells you if it's going in a circle or not?
No, I don't think that was what is happening. I don't think that you can make noise to drive the behavior of the cell in order to be persistent or oscillatory, or in 2D being circular or exploratory. That strongly depends on how fast the cells are able to build the footprint. If the cell doesn't secrete enough footprint, it cannot build the necessary substrate to keep moving in the same direction, and that is going to force the cell to repolarize. If the cell has a stronger secretion rate and is able to build this footprint as it goes, then this cell can start moving into new regions. And that cannot be controlled by noise. That is just controlled by basically the ability of cells to build the substrate.
You said 12 of the 150 are circular and the other 138 are of this type, right? That's what you told us.
Yes.
But there aren't any intermediate ones, one that start like this and then become circle. It's easy to split them into categories and that's it.
So you're thinking of on this one like a circle?
Yeah, you said there are 12 of one type and 138 of the other.
Yes.
There aren't any intermediate ones?
Sometimes yeah, okay. So we observe sometimes that some cells, for example, they will start like moving in circles, and at some point they start becoming explored.
So that makes it sound like noise, right? You're not changing the cell, you're changing the motion.
So what I would say to answer that is our model cannot explain that by noise. So it might be that something else is happening with the real cells, but our model doesn't predict that is going to happen with noise. and I don't think that's happened with noise. If something is happening to the cell, it might be that the cell is basically changing the way that interact with the environment in some way. So cells are living objects, so many things are happening at the same time. So this idea of assuming that the cell behaves in the same deterministic way all the time might not be correct always. So I wouldn't be surprised to see cells in the experiment that behave for sometimes in a way, and after some time they start behaving on another way. There are many things that happen to the cells as we are observing them.
Sorry, Emiliano, if I understood it right, it is the parameter of one of the parameters what is driving this transition, right, between behaviors.
Yeah, that is the secretion
The
rate.
deposition rate.
Yeah.
So it could be easily that cells, for any physiological reason, they are kind of in the transition and their physiological rate is fluctuating, right?
Yeah, that is one of the points that we want to raise here. So we cannot we cannot assure that, we cannot say yes, cells are doing that, but our data suggests that they could do that, and that could be a very interesting way of controlling their motility. And if you are, and that makes a sense from the evolutionary point of view, and if your cells has evolved in a way that they are set up very close to this transition, that then it's very easy for the cell to change one of these parameters, to change very rapidly the way that they behave. So you can change by making small changes in the way that you secrete your footprint, you can become exploratory or you can become confined. So that is a very powerful tool that the cell could have. But of course, we cannot say that that is happening, but we can speculate that that could be a possibility.
Do people know if this deposition rate is related to starvation or things like that? Because I would think that a way of switching between this kind of confined movement and exploratory is something like triggering foraging or things like that, or predation, or they have
It might be possible. I don't think that this is what happened in this experiment. I think in this experiment, cells are giving a media that they are quite happy about. But it could be that cells change or reprogramming themselves according to the situation they are facing. So that is also kind of the claim that I'm trying to do here, is that motility is going to depend strongly on the environmental conditions. And so the idea that cells, as they move, they're changing the environmental condition, and the changes in the environmental condition are changing the way that cells move. So this is a very dynamic process.
Do you understand why the critical K is different in 1D and 2D?
Why we have these transitions in different places?
Yes.
So this has to do, I think one of the reasons is, okay, so the answer is we have not a clear idea why this is happening. One of our what we think is going on is that this idea that the cell can repolarize, shift the polarity front easily in 2D than 1D. So it's just easier to the cell to switch direction and start moving into a new direction. On the other hand, for a cell, it's just much harder because it has to repolarize in the opposite direction. So that affects the way these repolarization events. On the other hand, there's something that has to do with the shape. So we made simulations changing the tension of the cell, so making the cell more elastic or less elastic, and that shift the position of this transition, but that shift the transition of this position in a non-monotonic way.
I see.
So it's not that simple to understand exactly what is triggering this transition in one case into the, but certainly it has to do with the shape of the cell.
Okay.
Also, something that is important, in 1D cells are more elongated, in 2D cells are more rounded. So that's something I forgot to mention, but you can check that on the movies. So you can see these cells are not as elongated as they were before. And the reason for that is because they can have a larger front.
Emiliano.
Yeah.
Your rate kappa of secretion is fixed in time, right?
Yes.
If you oscillate that, fluctuate that stochastically, then you could have transitions between the two dynamics, right? So the intermittent behavior that Nathan mentioned.
Yeah, that could be true. Oh, and if by that he meant noise, yes, that could be perfectly reasonable to think about, that the noise is in the secretion rate, and so that noise makes you sometimes be on one type of motility and sometimes on another type of motility. But these behaviors, they are just not fluctuating kind of very fast. So that noise should be changed with a very long time scale, so that you can have very long time scale where you observe your circular trajectories and then your exploratory trajectory. So
Yeah,
if your noise is changing all the time, you will see something that doesn't look at all like a circular trajectory, and doesn't look at all like an exploratory, something that is switching all the time between these.
For the motion of bacteria, the reorientation rate has a memory of 20 seconds, more or less. So it makes sense to think of a rate that has a memory, a fluctuating stochastic memory of.
Yeah, 20 seconds. Yeah, 20 seconds here is nothing. So these eukaryotic cells are really, really small, and the time scale here are hours. So these movies, they have probably like 12 hours, 14 hours, or something like that.
Yeah, but you're talking about crawling cells, right?
Crawling cells, yes.
Yeah. In the case I mentioned, it was like swimming, right? So
yeah, so bacteria is a different story. Bacteria is much faster, it's much smaller, and the time scales involved in the bacteria are much shorter.
Thank you.
Yeah. So as I was saying, okay, so is there any other question, or should I continue with the mean square displacement? Okay, so here on the left, we have the mean square displacement for the simulations, and here on the right, we have the mean square displacement for the experiments. The red curves are control experiments where they condition the substrate. So basically, they place at high density the substrate. So to be covered full by cells, they leave the cell for some time, and then they remove the cells. In that way, that substrate should be full of footprints. Okay, and then they take that conditioned substrate and they place new fresh cells over that. And what they observe is basically that in that case, the cells behave kind of more in this persistent active random walker way. So that you observe the T square at the beginning, the ballistic phase, and then you see the diffusing phase at longer time. For the experimental condition where there is no conditioned substrate, what they observe is that you have something that is kind of ballistic, but in the long term, they don't observe diffusivity, but something that has an exponent that is between 1 and 2/3. So something that is moving kind of in a subdiffusive way. In our simulations, we can see something similar. So for example, in the circular motion, so being below the transition rate in 2D, we see that our mean square displacement grows as 2/3. So this is a subdiffusive behavior. When we increase by a lot the secretion rate, we start seeing some simulations that looks like the typical ballistic diffusing regime. But something in between happened. This is the secretion rate that is kind of slightly above the transition. We see something that looks so diffusive with the exponent that is between 1 and 2/3, kind of like in the experiment. So this is kind of also suggesting that these cells are living in close to the transition point, slightly above the transition point. And okay, So with that, that concludes the first part of the talk. And I show you that we can build a model where cells secrete and interact with the footprint, and that means that interaction can produce oscillation, but it can also produce persistent motion, and we can control that motility behavior by just changing how fast the cells secrete the footprint. In 2D, we observed expanded circular and exploratory behavior, and we were able to observe those type of motility behaviors on experiment. And also our model suggests that cells, real cells, should be slightly above the transition. And be happy to take question about this, and let me know if you want me to continue over the part 2, but it's free all night. I don't mind, but I don't know.
I would like to hear more, but I just have a question. If you have two cells, can a cell distinguish its footprint from another footprint?
That's a good question. In principle, no. Okay, so in the experiment, they observed that they don't. So basically, with this conditioned substrate, they observed that cells can respond in the same way to other cellular footprint than to the cellular footprint that they deposit itself. So what happens basically when they condition this substrate placing the footprint of other cells, and when they place a new cell over there, that cell, instead of oscillating, is going just to move persistently. So that cell, it doesn't care if the footprint is from its own or is another, is secreted by other cells. In our model, we don't make any distinction, but in principle, we could.
Okay.
Any other question?
I have another question. Emiliano, is it possible to derive an equation of motion for the center of mass of the cell using the phase field model?
I would say that I wouldn't know how to do that. It doesn't mean that it's not possible. It's just I wouldn't know how to do that.
Yeah, probably the problem is to integrate the phase field equation, because in principle you can integrate R deposition using phi as a weight.
Yeah, so you could do that, but the problem. So the reason that people use a phase field is because you have to deal. If you try to go that direction, you have to deal with complex boundary conditions. So the phase field allows you to deal with complex boundary conditions in a very natural way without any complexity. Okay, so you can see here how much depends the motility of the cell on the shape of the cell, and where the footprint is going to be placed. That also depends on the shape. So there are a lot of things that depends strongly on the shape of the cell, and those are boundaries that are continually changing. And if you have to track that over time, it's a really hard problem to do. It doesn't mean that you cannot solve it. I think it's just probably is going to be very hard.
Thank you.
Any other questions?
Okay, so just to be clear, should I continue?
Yeah, please.
Okay. So now in part 2, I'm going to shift organism. I'm not going to talk anymore about eukaryotic cell. Now I'm going to talk about bacteria. And as I mentioned before, bacteria are much smaller than eukaryotic cells. They don't crawl, but they swim, and they swim much faster than they move much faster than the eukaryotic cells. And what we want to understand here is the bacterial swimming close to surfaces. And the reason that we want to understand bacteria swimming close to the surface is because this is very relevant in the infection process. So as I mentioned before, Salmonella, for example, has to travel from the gut lumen to the epithelial wall. And once that it arrives to these walls, it has to kind of explore the wall surface to try to locate a site to anchor and start infection. So we wonder how swimming near a closed a surface affect the way that bacteria swim. So I think most people is familiar with the idea of bacteria performing run and tumble. So when bacteria is far from surfaces and is on the ball, they usually move in this run and tumble fashion, where the bacteria is moving in one direction for some time, that is called a run, and at some point it's going to tumble, change the direction, and then keep moving in one direction again. So this is run, tumble, and run, tumble. Okay, and it is well known that cells are able to use this tumbling as a way of performing chemotaxis. So basically, bacteria can regulate the tumbling frequency to bias its motion towards food sources, for example. But what happens when bacteria is close to the surface? So when a bacteria is close to the surface, it has been observed that instead of moving in this run and tumble, now what they observe is that they swim in circles. As you can see in this trajectory, this is a real trajectory of cells of bacteria on a surface. And it has been also observed that these bacteria stay for a very long time over the surface. So this is suggesting that the surface kind of track bacteria towards it. And also that these trajectories are very smooth and no tumbling are being observed during bacteria close to the surface. And if we don't have tumbling when the bacteria is close to the surface, now we can ask, okay, so how bacteria make chemotaxis if they cannot regulate tumbling? So to try to answer this question, we performed some experiments where we place a Escherichia coli over this petri dish, and then we just observe the bottom of the petri dish. And this is what we found over here. So this is what has been reported before. So basically a cell that is moving in circles, a small circle, so we don't see any tumbling. And we observe that this cell is remaining over the surface for a very long time. But we also observe something else. We observe this type of trajectories where the cell is moving in a circle and then stopping, changing the direction, and start moving again in a circle. And here again. So we have this event where the cell is moving, stopping, changing direction, moving, stopping, changing direction. So this is very similar to the idea of the run and tumble. So now instead of run and tumble, it's kind of chiral run and stop events. Okay. So we wanted to characterize these stop events. So for that, we measured the bacterial speed, and then we define a threshold. So when the speed of bacteria was below a certain value, we would assume that the cell stopped. And when it is above, we would assume that the cell is moving, is running. Okay, so this allows us to compute, for example, quantities of the change of direction during the stop event, and we can write the probability distribution of this change of directions. And we can also compute the time, the duration of these events. And with this, we can build this time probability distribution of the events. So something that was a bit unexpected for us is what we observe here. So for the running time distribution, so this red curve, this is what we were expecting. So we observed that this is well fit with a single exponential. So that is compatible with the idea that there is a single time scale in this problem, and it's probably is associated with the idea that there is a constant rate of becoming running become stop from the running. But when we see the stopping time distribution, now we see something that doesn't fit very well with one exponent, but it actually fits very well with two exponents. So this is suggesting that there is no single timescale here, but there are multiple, at least two. So this is suggesting that our stop event have multiple states. So we propose a very simple, what we could manage to make the simple model to describe this time distribution, in which we consider that the running state is described by a single state, but the stopping state are described by two different states. And then we have this transition that we plot over here. So by doing this, now we can write our stopping time distribution, and since there is two state and two rate, now we can find this probability distribution, time distribution that has two exponents, and then we can fit our data with this expression. And when we do that, we find the two time scales over here. One is really fast, that is 0.2 seconds, and the other one that is 5 seconds. And now we can associate this time scale with different physical phenomena that is happening. So this 0.2 is consistent with the fluctuations in the flagellar motors. But this 5 seconds time scale is much larger, and we believe it is associated with adhesion. And let me show you this movie to show you that we actually observe adhesion here. And you can see that the adhesion, because it manifests here in the case of tethered movement. So basically, one flagella, the flagellum gets stuck to the surface, and the cell is going to rotate kind of in a circle. And you will see it now. Okay, so we have now our trajectory. So we analyze the trajectory. We can define, okay, so again, the stopping event by being below the threshold, but we can also measure the curvature of the trajectory. And if the curvature of the trajectory is above a certain threshold that is associated with the curvature, the radius of curvature of a tethered event, we can see that this is a transition associated to the fluctuation of the flagellar motor. So this is not associated with adhesion. But if the curvature is below a value, that means that it's probably something that looks a lot like a tethered movement. And then we can associate that with the adhesion state. So basically, now we have a way of identifying when the cell is in state 0 and state 1 by doing this. And then by doing this, we can compute the probability, the rate of transition between all these different states. Questions so far?
So by
Okay.
adhesion,
Yeah.
sorry, by adhesion you mean attraction to the surface, which could be either of electrostatic origin or hydrodynamical origin, right?
Attraction to the surface is happening all the time, and this is happening by hydrodynamics. So this is why the cell is the bacteria is staying close to the surface. By adhesion, we mean physical adhesion of the bacteria to the substrate. And as I mentioned before, this is associated with the idea of the tether. So when you see the cell that the bacteria that is moving these circles, that means that the flagella got attached to the surface, and since the flagella is rotating, that makes the body of bacteria to rotate in a circle. So it's like you catch the bacteria from its tails over the surface.
Mm-hmm. Thank you.
Okay, so now we are able to define our states and measure the different change of probability of change from one state to the other. But now we want to add some motility to this. So for that, we are going to consider our bacteria as a chiral active particle. So basically, this is described by this equation over here. So we have the typical active particle equation over here, where the cell is moving with a given speed and a director, and the direction of that director, or we can think about the orientation of the bacteria, is given by this equation. That has some noise that has to do with thermal angular noise with some diffusivity d theta. We also add, in this case, this angular speed to account for the fact that cells are moving in circles when they are over the substrate. Okay, but of course, this only happens when cells are on the state 2, when they are in the running state. When they are in the stop state, of course, they are not moving and nothing is happening with their equation of motion. And then what we assume is that whenever this transition takes place from a stop event to a running state, we draw a change in the orientation of the bacteria from a probability distribution that was measured from the experiment. Okay, so this happened kind of like instantaneously when the transition takes place. So we can write the Fokker-Planck equation for this model, and this is a bit involved to solve this, but the main idea is basically that we have to assume a simple shape for these distributions, that these are basically distribution of how for these angles, the change in direction when the transition takes place. And for that, we assume like a top hat distribution, so something that is constant between some values. And that allows us to simplify this equation. Then what we do is we make a Taylor approximation up to second moment. And then we we Fourier transform, and then we can get the Asian value and Asian vector of this. And then we use the Taylor Kubo formula to compute the second moment of X. And by doing that, and then taking time to infinity, we can find an expression for the diffusion coefficient. That is this kind of I was going to say ugly expression, but it's not ugly at all. I think it's quite simple after all. But yeah, we can find this diffusion equation, expression for the diffusion of the bacteria. So now we want to explore what this equation is telling us. So for that, we are going to play around with some of the parameters of our model. And for that, we choose the K2 rate, that basically the rate from being on state 2 to state 0. So this is basically the rate of stopping, or how frequent the bacteria stop. And then we can plot the diffusion coefficient as a function of this stopping frequency parameter. And what we observed that is surprising, was kind of surprising at that moment, is that this bacteria can increase diffusivity by increasing the frequency of stopping. So this is kind of a very unintuitive. This bacteria is going to increase diffusivity by stopping. Okay, and I will go in a bit to why is that. But first, let's try to understand the shape of this curve. So if we go to k to 0 to infinity, so the stopping frequency that too large, that means that the cell is stopped all the time. So immediately that it goes to the running state, it's going to go back to stop. So this cell is not moving, so this is why the diffusion coefficient goes to 0. On the other hand, if the stopping frequency is too small, that means that this bacteria never stops. So it just keep moving as a chiral active particle. And if that is the case, then we have the diffusion coefficient that takes this value. And this is consistent with the experimental trajectory that we observe here. And in the middle, what we have is this intermediate case where the bacteria is stopping. And why is maximizing or increasing the diffusivity is because exploring circles is not very efficient. So that means that you are kind of basically always trapped in the same region and becoming again and again and again visiting all the time the same regions. So this bacteria is kind of very inefficient explorer. But by stopping and restarting or resuming motion in a new direction, now you are able to break these circular trajectories and start moving into new trajectories. And this is what we observe in this picture, and we can see how this bacteria, much shorter time scale, it was able to explore a much larger region than this bacteria over here that never stopped. So lastly, what we did, we went back to the experiment. We measured what was the stopping frequency from the experiments, and what we found was that it was very close to the optimal value of our theoretical curve, suggesting somehow that this bacteria evolved to maximize their diffusivity over surfaces. And this is just the last concluded remarks, is that we can write our diffusion equation as the one that I showed you before that was a bit much more complex. We can simplify it like this, and where we have this d theta effective, and we can think about this theta effective as the contribution of different type of changing direction sources. And those different changing direction sources can be from adhesion, for example, as we observe in our case, they can be from tumbling, they can be from collisions, or they can be from something else. So now we have a way of increasing our diffusivity, angular diffusivity, by adding these new processes over here. And this is also what would happen if we add obstacles. So this is something that we are interested in explore now, the idea of adding obstacles and how that affects the bacterial motility. And this is, again, a bit counterintuitive in the sense that when we start thinking about obstacles, it was because we were trying to think a way of impeding the cell to reach target cells. So we thought, okay, if we add obstacles to the medium, that would make cells to be less effective in reaching the target cells to start infections. But that's actually not what happened. What we see that could happen is that by just adding obstacle, we're increasing the diffusivity of the cell, and that would increase the infection capacity of these cells, of the bacteria. And the last comment over here would be that this idea of these extra sources, this could be a potential mechanism for bacteria to recover chemotaxis over the surface. So if bacteria is able to control the adhesion frequency, what you could define as tumbling into the collision, they won't be able to control. But if they can, for example, control the stopping frequency or the adhesion frequency, then they will have a way to recover chemotaxis over surfaces. And with that, I I got to the end of the talk, and so this is the acknowledgement. So the work I done over with the cellular footprint has been done with Professor Brian Camley and our collaborators Joseph D'Alessandro, Benoladu, and the work with the bacteria has been done with Fernando Peruani and Stefan Otte in University of Nice. They changed the name to Côte d'Azur. And these are Dorota Cheruca and Rodolphe Ponte, the people that made the experiment for us. And with that, I would like to thank you and take any question you have.
Okay, thanks very much. Any question?
I mean, just a very basic question. So this field of active matter is a very big field, right? So what exactly is your approach? How do you distinguish yourself from everyone else that works on these random walks with small objects and so on? What is for a non-expert, is there something that summarizes your program and that distinguishes your program from a standard?
So I would say that what differentiates me mostly is I always been working close to real biological system to try to understand more the biological problem than the physical problem behind. I know there is a huge community of people trying to understand random worker processes or the properties of these active particles, for example. But so far, my way of working has been trying to attack or try to understand a biological question, and from there try to understand the physics behind that question. And that involves also working much more closely with experimental data. So I think one thing that differentiate me from other people that is working on this subject is that I do a lot of data analysis. It's very important how to try to understand how to get reliable data or information from experimental data. So many of these work has been basically done by just trying to understand what we are observing from the experiments, as you can see over here.
Yeah.
So the other thing that I think that distinguishes me is that I don't think there is many people trying to understand, at least in the way that I'm trying to do, the idea of the interplay between the motility and the environment. I think that most people have focused so far on understanding how motility affects the environment or how the environment affects the motility, but they haven't been put these two things together to try to understand them as a whole thing. And I think that this bacterial infection process and also the cellular footprint, these are very nice example of how this interplay takes place. So this idea of the obstacles is a very nice idea of the infection. So we use yeast as obstacle for the bacteria. And what happens is that after some time, bacteria start colliding with the yeast, and they are going to, so the yeast are not fixed obstacles in the medium, so they can be displaced, and bacteria in each collision start displacing them. So after sometimes you will see all the obstacles, all the yeast accumulates in some regions, and that is a very dynamical process, and that changed a lot the topology of the environment where the cells are moving. So the infection capacity or the searching times for the bacteria to reach a target cell, it's going to change or evolve within time. So we are observing a process that is evolving with time, and that evolution of time has to do actually with the way that bacteria move and the way that bacteria move. also is going to depend on how this rearrangement of the outcells takes place. And with the footprint, we have the same situation. Cells need to use the footprint to be able to move. And this is very interesting from the point of view as a guiding mechanism, because this would allow cells to have their own control of basically how they migrate. So this is a guiding mechanism that they are building by themselves. So they have their own way of building a mechanism that basically allows at some point to be confined or to stay moving over a confined region, or at some point say, okay, I'm going to start exploring and start moving into a new region. And those are important changes that has important consequences in real life. So for example, that's the case of tumor cells. So tumor cells, for some reason, at some point they decide, oh, they're going to start moving away from the tumor. And I was going to, okay, so and the idea of the footprint is also like they can use footprint to guide other cells. So you can have a leader cell that deposits some footprint so other cells can follow them. And this footprint can be can change over time. So this is a very dynamic and process that is going to evolve with time for sure.
Okay. I see. So my feeling from the outside was that everyone in this active matter was very closely connected to experiments because of the scales, but that's not true. Some people do much more of the mathematical modeling, some people do just the experiment, and there are a few people like you that are really in the interface between the two.
And there are many people that do experiments, but they might be doing experiments to try to understand their mathematical models. So they might experiment probably specifically to try to answer the question, a physical question behind. All of this has been more focused on trying to understand a biological question behind. So cell migration, cell motility, infection capacity, and so on.
And just one more question, and then I think I have to run. Under these experiments, are they done? Are you involved in also helping do the experiments or suggest what people measure? Or
so the footprint experiment, we were involved because this was a work that was already published when we started working on that. We might start moving in that direction, that we start suggesting new experiments so we can try new things on our model. But on the experiments of bacteria, I took place. I made a few experiments. They are not the ones that we observed that I showed you recently, but I made a few experiments. And at some point, yes, we were defining altogether which were the next experiments to be done.
interesting. Very nice. Thank you. Thanks for a nice talk.
Might have, let's say, related question. I mean, in the talk you showed many, let's say, numerical simulations and so on. You are were you in charge of the numerical simulation or you do? Can I ask you what kind of algorithm and what kind of language do you use for the numerical simulations?
Yes. So this. I have become lazy with the time, so now I'm using mostly Python.
Mm-hmm.
So this phase field model was done basically with Python, using Numba to kind of accelerate.
Mm-hmm.
For the simulation of the active chiral particles, so these simulations, so the circle here are simulations. That were done in C, basically. I did that on C, so that meant that that was much faster, and I was able to make much more data. But yeah, I'm the one doing the numerical simulation and writing the code and
I see.
also analyzing the data. Yeah.
Okay.
Any other questions? Okay, so Emiliano, just to clarify, it's going to be Thursday, 10:30.
Yes.
Okay.
Yes.
So thanks very much, and look forward to your next talk.
Okay. Thank you, and see you on Thursday.
Bye-bye.
Have a nice day. Bye.
Bye.